Good day to all. We, the second group, will now be presenting about cell-mediated immunity. Before we start, here is a list of our group members who will be presenting today. The presentation will be divided into two parts. In the first part, we will be introducing the cell-mediated immunity, and we shall go over the main players in the system and take a look at the additional components that we think you should know. In the second part, we will learn about the pathways and mechanisms. We will take a look at how all those components react with one another in order to bring about an immune response, and what exactly those immune responses that we're talking about are. So, let's get started.
What is cell-mediated immunity? It is an immune response that is produced by direct action of immune cells, which is in these cases, T cells is the main immune cells. This response doesn't involve antibodies. And why is it called cell-mediated? It actually refers to the action of T cells themselves that will latch onto the pathogen's antigen and initiate the destruction.
The cell-mediated immunity occurs when there are the infected cells in the body, and it involves the T helper cells, which recognize the antigens that are presented by the macrophages, and then it releases the cytokines and chemokines to start the process. This cell-mediated immunity system, it also involves in the production of the antigen-specific cytotoxins, T lymphocytes, and any of the other white blood cells.
So now, we will look at the major histocompatibility complex, which is known as MHC. It is a group of genes that code for proteins found on the surface of cells that help the immune system recognize the foreign substance. There are two types of MHC, which are Class 1 MHC and Class 2 MHC. Class 1 MHC can be found on the surface of all nucleated cells in mammals. It presents antigens to cytotoxic T cells and is responsible for the clearance of endogenous antigens. Class 2 MHC can be found on antigen-presenting cells such as macrophages, dendritic cells, and B cells. It presents antigen to helper T cells and responsible for the clearance of exogenous antigens.
Now I will talk about the types of T cells. As you can see in the picture, the T cell is presented with an antigen with a complementary shaped receptor. This then stimulates it to form into four different types of T cells with helper T cells, memory T cells, suppressor T cells, and cytotoxic T cells. Helper T cell exist other white blood cells in immunologic processes, while the memory T cell are the subset of T cells that respond to a previously encountered antigen. Suppressor T cells actively suppress the activation of the immune system and prevent the pathological self-reactivity. The cytotoxic T cell attack foreign cells or body cells that are infected by viruses. Now, I will pass to Wan Xing to talk about the structural difference of the T cells.
So next, I will discuss about the structural differences of T cell. The first one is the helper T cell, and the CD4 is the surface CD molecule, and the antigen-presenting cell presenting the antigen associated with the MHC2 is the activation of the helper T cell, and the function of it is to assist other white blood cells in immunology processes. The second cell is the regulatory T cell, and the CD4 is the surface CD molecule, and the activation of it is the antigen-presenting cell presenting antigen associated with the MHC2, and it involved in peripheral tolerance and prevention of autoimmune responses. Third one is the cytotoxic T cell. The surface CD molecule is CD8, antigen-presenting cell or infected nucleated cell presenting antigen associated with MHC1 molecule. And the function of the cytotoxic T cell is to destroy infected target cells such as virus infected and the tumor cell. So the last T cell is the memory T cell. The surface CD molecule is either CD4 plus or CD8 plus, is depending on the type of the antigen encountered. So the activation of the memory T cell is to respond to antigen presented on other antigen presenting cell, including the resting B cell. And the function of it is to subset infection as well as potentially cancer fighting T cell.
Alright, now we will take a look at how antigens leads to the activation of T cells. Let's take a look at the helper T cells. You might remember from the first group that phagocytes are capable of breaking down antigens into smaller fragments and display them on MHC2 molecules. They present this antigen, and that's why we call them antigen-presenting cells. This complex of antigen with MHC2 molecule is needed to stimulate the activation of our helper T cells, and you can see that helper T cells actually have a receptor to bind to this complex. Another protein called the CD4 is not shown here, assists in this binding process. Besides, full activation of T cells requires co-stimulation from other molecules on the APC. Only when the first two steps are done, the helper T cells can now be fully activated and undergo clonal selection, which will form active helper T cells and memory helper T cells. This is a picture showing the CD4 molecules mentioned just now that assist in the binding process. And this picture shows you that the full activation of helper T cell is not just a simple binding between the receptor with the MHC2 antigen complex. It is also involved tons of other protein, but for now, don't worry about it. Now, an activated helper T is capable of releasing interleukin 2, which is a very important cytokine to charge the immune system up. It can either bind back to the helper T cells that secretes it, further promotes proliferation, and further release more interleukin 2, or it can bind to other cells of the immune system listed here and really kickstart the immune system.
Activation of cytotoxic T cells. Activation of cytotoxic T cells consists of two steps. First step is cytotoxic T cell will recognize the foreign antigen by binding of the TCR and CP8 protein that locate on the surface of cytotoxic T cell with the MHC1 molecules on the surface of infected cell. Second step is where the co-stimulation occurs, which co-stimulation by interleukin-2 and other cytokines that produce by active helper T cells that have bound to the same copies of antigen. By having both steps, cause the activation of cytotoxic T cells. After the activation of cytotoxic T cells, cytotoxic T cells will undergo the clonal selection, which produce more clone of cytotoxic T cells, which consists of active cytotoxic T cells and memory cytotoxic T cells.
Binding of antigen by infected cells to the receptor activates the lymphocytes, causing proliferation and differentiation of cytotoxic T cells. This is what we call clonal selection, where antigen selects particular lymphocyte for clonal expansion. This differentiation will produce active cytotoxic T cells and memory cytotoxic T cells. Active cytotoxic T cells will recognize and attack the infected body cells with one particular type of microbe. For memory T cells, it is a long-lived cells that offers a fast response to the previous infection, the same infection to be exact, by proliferating and differentiating into more active cytotoxic T cell and memory cytotoxic T cell.
Again, what do cytotoxic T cells do? It's similar like the soldiers as main characteristic in this killing action that mediates the immune response. The elimination of pathogens inside cells happen by these soldiers recognize enemies by specific antigen on MHC1. Once a cytotoxic T cell recognizes a cell that displays foreign antigen, it kills cells via a variety of proteins and enzymes such as granzymes, porphyrin, granulysin, and lymphotoxin. There are two mechanisms of killing action. First is apoptosis induction and secondly direct attack on a target cell's membrane. In this apoptosis induction, porphyrin will form a pore and delivering of granzymes, which proteins digesting enzyme that trigger apoptosis. The released microbes are then killed by phagocytes. This figure just to show how T cells killing action which have been explained in the previous slide. The other mechanism for killing action is by directly attack on the target membrane of the infected cells. These cells bind to the infected body cells and release two proteins from their granules. First is perforin, which inserts into plasma membrane of target cells and creates channel in the membrane. As a result, ECF flows into the target cells and cytolysis occurs. Second protein that releases is granulysin, which creates holes in target plasma membrane. Alternatively, cytotoxic T cells may also releasing another toxic molecule called lymphotoxin, which then activates enzyme in the target cells. This enzyme causes the target cells DNA to fragment and the cells dies. This figure also shows how this mechanism works in previous slide explanation.
To sum up, cell-mediated immunity are called so due to the action of the T cells that latch onto infected cells in order to kill them. We have seen the cells that are responsible in this system, mainly the helper T cells and the cytotoxic T cells. By now, you should be able to differentiate between the two. We also have went through the activation pathway of the T cells, which begins with the activation of helper T cells and then leading to the activation of cytotoxic T cells for killing. Hopefully, through this presentation, you also realize that MHC molecules play an important role in this pathway, and different MHC complexes may stimulate different cells. We also emphasize on the concept of co-stimulation, which is something quite new to us and is quite a unique feature for the immune system. Lastly, we'll also take a look at the different enzymes and the proteins used by T cells for killing, such as perforin, granzyme, lymphotoxin, and so on. With that, I conclude our presentation for today. Thank you for listening.